Yes, Carl Brunius at Chalmers University of Technology, as mentioned, Department of Life Sciences. We just changed name from Biology and Biological Engineering, and at the Division of Food Science and Nutrition. So you will understand that I have a particular interest then in dietary exposures and their relation to health. And we have, I would say, semi-large throughput. We run our analysis at the Chalmers mass spectrometry infrastructure, where I'm also the scientific advisor, and we have roughly 30,000 injections per year on some dedicated instruments. So it's probably not the scale of biobanking for that kind of scale, but still a lot of samples. And we have experienced some problems with data quality over the years that we have tried to address with this package that we call QualiMon LaMa, quality monitoring with landmark features. Yes, and as I mentioned, in the field of basically food and health, the general research area is to integrate exposures to health. And by exposures, we look predominantly at diet, but diet is also largely interacting with microbiota, and that's also one of the most important vectors for environmental pollutants. So how do we integrate these multiple exposures on multiple health outcomes? And a strong research direction that I have is to use the molecular regulation frequently covered by metabolomics, but also proteomics and other omics layers can be interesting here, and use that as the sort of common language for both exposures and outcomes to try to make mechanistic research and molecular epidemiology. But of course, we need metabolomics data, and we have a team in computational metabolomics. I'm not the head of all of these guys. It's just people that I find myself working with a lot. So everything from people who are more specified in metabolite identification or application in particular areas, immunomaturation or dynamic data analysis. So that this guy is working more on pure math and so a lot of different aspects of computational metabolomics, all the way from generation of metabolomics data, pre-processing on the top line, actual data analysis, and post-processing in terms of identification. So development of a lot of procedures. We've been in this field since approximately 2014, and over the years, we've developed some algorithms of our own for drift correction, data analysis, molecular epidemiology, cleaning out false peaks from XCMS-generated data. Identification, we're working on that. So that's current work together with the latest development of the quality monitoring package. The rationale for that is, of course, that working with high resolution LCMS instruments, they're really sensitive, and that's why we love working with them. But they're not very stable over time, and we have all of these quality problems that are really impossible to avoid. And we've had several procedures in place to examine quality, and mostly that has been manual work by operators to basically extract information about a handful between 5 and 10 known peaks, normally highly expressed in, for example, plasma. And they look at those peaks with a few metrics and ascertain whether or not the quality is good. So one problem with that is that it's very time inefficient. It takes a lot of operator resources to do that. And sometimes we didn't get responses about batch quality until after a few batches had been run. But more importantly, we found that it wasn't fit for purpose. Several times we had indications from this manual assessment that sample quality for the batch was, or data quality for the batch was good, when in fact, when we start to do the peak picking procedures, we found that it was horrible. So a handful of highly expressed features is not representative of the quality of the data from an untargeted point of view. So we borrowed some ideas actually from Jan Stanstrup from what he held a presentation, I think it was in 2017 at the Metabolomics conference in Melbourne, and we started talking about procedures. So we had some thoughts and decided to to try our hand at developing this algorithm. So we're using landmark features that I'm going to explain in just a minute. And what happens is we discover problems with particular injections 2 minutes after it's completed, and it's fully automated. And it's all written in open source based on R with a shiny interface, and you're using SQL, the databasing. So it's basically plug and play for anyone to use. So what are the landmarks? Well, we had the rationale for it. Those are features that are virtually omnipresent. Basically, they should be found in virtually any sample of a given sample matrix. That is plasma, for example, or urine or adipose tissue, but it's also specific to the analytical mode. So basically, we have one of these maps for every sample matrix and every analytical mode. So this, I think this is blood plasma, reverse phase positive, but I may remember wrong. They look very similar between the different modes. The rule set for identifying these landmarks, and we work with approximately between 100 and 200 of these landmarks for any given mode, is that there should be low missingness. Virtually, it should be possible to find in any injection of a certain sample matrix. It should be in high intensity, so there is no risk of not picking it up. It should have no immediate neighbors in retention time and mass to charge ratio, so we cannot mistake it for anything else. And preferably, it should cover the RTMZ space so that we don't have any blind spots in the map. And how to do this? Well, luckily in this package and this software, it comes shipped with a tutorial which explains all the various steps that you take. Plus, there's a built-in wizard where you can load in data sets, so you can identify these from your own historical data on your platform. And there's an optimization algorithm running under the hood to help you identify a set of landmarks that you can work with. The procedure then, what happens in a total flowchart is that first I need to mention that it requires at present at least a uniform naming strategy, which we have where we name our samples by experiment date, batch, chromatography, polarity, sample name, and injection number in the sequence. So all the metadata that is required for databasing is present in the name, so we can check at any given time point that where an injection belongs. So that's, I would say, crucial to this. Moreover, from a data security perspective, all of these processing is done on a processing computer, which is physically separated from the instrument computer with only read access. So we have no chance of modifying what happens on the instrument computer. We've had bad luck with that previously before we started to clean up those procedures, that sometimes we have data being destroyed or corrupted. But we basically have a monitoring of the location of the instrument computer where raw files are being generated. And once we find that there's a new file, we monitor it for its file size until it stops increasing in file size. Then we know it's complete. And then we copy it to the processing computer. Of course, this can be done on the same computer, but I would strongly advise to separate them. That means we have automated backup of the files, which is very nice. So we don't have to depend on any batch-wise procedures for that, or not remembering or anything, but it's all on the fly. It's automatically converted to MZML and then read into R using XCMS. Using standardized parameters so we can compare injections between experiments and between batches and whatnot. So from each injection, we extract some sample level data and some landmark level data. And I will go through those particular metrics that we're using in just a minute. We store that in an SQL database and compare this sample to all of the other samples that were generated in this experiment to figure out whether it behaves differently or not. And then we have both what we refer to as relative test, which is we compare it to the other injections, and we also have absolute test, whether or not it performs bad according to hard threshold values that we know about our system. For example, the number of peaks that we should be able to find in a particular sample. So the metrics that we use to base our decisions on, it's on the level of the injection, as on a global level of the injection. Those are the number of peaks that we pick using these standard parameters. And you can see a visualization, and this is 1, 2, 3, 4, 5, 6 batches. And you see that number of peaks it reduces over time due to buildup of dirt in the interface, for example. This is just extracted from historical data. Nowadays we stop way before we come to this kind of situation. The IPO score, which if you're not familiar with that, is isotopolog. So it basically checks, it's representative more of stable peaks than the actual number of peaks. It's total ion current, the number of landmarks that we find per sample. So we can see it actually finds a lot of these landmarks in the sample, except for some injections where it's a bit strange. And then we can examine those samples for what went wrong. There's, for every of these landmarks that we find, in this case, it's about 120, 130 landmarks. We also extract retention time, area, height, noise, signal to noise ratio, and etc. All of these metrics, so combined, we have for every injection something in the order of 1,000 different metrics per injection that we base our decision of sample quality on. So, and we extract this using a package that we developed together with some guys at Uppsala University, which was very nice. So this is the main look at of the software. So as I mentioned, it's written in R, and this is a shiny, GUI interface. So it's like web browser based, and we can see that this particular experiment is contains 11 batches with 879 samples, and this is historical data where this procedure was not in place. In fact, the manual assessment indicated that all of these samples were of sufficiently high quality, but we can see that the number of peaks went from 11,000 down to approximately 2,000 for some of the batches, indicating that manual assessment is not necessarily fit for purpose. So for these injection level metrics, we have soft and hard limits. If it's below 5,000, then we give a warning. So we penalize that to some kind of penalty score. If it's below 2,000 peaks in a sample, then we get a hard warning. And the same thing goes for all of these different metrics. For the landmark level metrics, it's a little bit more complex because we have so many of them. So basically on the diagonal, you see every new injection as it's being made. So time-wise, you're going to the right, we see what happens with every new injection. On the y-axis, we actually recalculate these scores for one sample. So you see this batch here, start of a new batch, its light color means it's probably a bad sample. So it says it's bad, but as we add more sample, we find that, for example, the number of peaks, it can be a batch effect that just indicates that the sample could be bad. But as we generate more and more samples, we may find that in fact it's not bad. It was just early stages of batch effect. So we continuously recalculate the quality of each injection over time. I think there's someone with a microphone on. But this is basically the main view, and we can show various plots of intensity outliers, retention time outliers, and etc. And then we can combine all of this into the overall status plot to give us an indication of what samples are good or bad. And you will find that these samples here are pretty much all of them light, indicating bad, and they correspond to these batches. There's a set of samples over here corresponding to those, and the final batch here, which is bad. And over time, as we have worked with this, we can pretty much identify from just eyeballing this status plot and going into some of these particular guys to see, does this relate to sample preparation? Does it relate to leakage in the system? What does it relate to? And if there is a warning for a sample, then we have real-time warnings on Slack, which goes directly to our operators to say that there is something happening. So they can just run down to the machine and confirm that it looks like there is some kind of malfunctioning, that there's a leakage somewhere, for example. So It's kind of complex. There's a lot of different modules in this, but luckily there's written tutorial which explains all the steps in setting this up, and this wizard will really help you a lot with this. So you go through step by step. Where are the folders generated? And if we separate from instrument and processing computer, we just choose a network folder. Then we can, where do we want to back them up? Where do we want to store the mz and miles and etc. So try to chunk it up into easily digestible pieces to help you set this up for yourself. So I hope to have shown you that we have implemented and arrived at the automated log quality monitoring, which also solves a problem of automated data backup. We get real-time warnings. It takes approximately 2 minutes for a sample to go through all the calculations after the injection is finished. We get real-time warnings on Slack if there's something fishy going on. It's vendor agnostic since we work with file conversions and mz and user-friendly. It's always a question mark around that one because, of course, it's complicated to write software, but we worked as much as we can on tutorials on wizards. There are some things that we are in the process of implementing, for example, switching between sample matrices, because the config file right now, you need to specify if it's plasma or urine or feces, whatever it is that you're working with, because landmarks are going to differ between matrices. We're working on re-implementing some of the databasing stuff so we can get automatic switching of sample matrices according to the file name. Logging temperature and back pressure is proving to be very difficult when working with mz files. So we're trying to implement ways to scrape that off the data, but yeah, hopefully getting there. MS2 is on the wish list, but we have no real means to prioritize that at the moment, but that's on the to-do list and then other things on the wish list. But I cannot tell you how much time this has saved us and how much resources in that we can abort sample sequences immediately from an automated point of view, basically, to not waste samples, not waste time, not waste solvents. So it gives us a much more resource efficient workflow. If there was time, I would have told you about some of the other algorithm developments that we have in the pipeline, but I think I should just stop here.